So this is Nathan Friedrich. He is a student of Dr. Barton's at University of Alabama, doing research in molecular biology. So to start it off, Nathan, I'll ask you, how do you feel about molecular biology?
I have strong and very unbiased opinions about molecular biology. It is clearly the best science. Joking aside, I think molecular biology is really neat. Just the entire concept of DNA as like a molecule that you can analyze using computers. Like it's biology, but it's also, I don't know, sort of like computer code sometimes. Molecular biology is a good science.
Okay. So you do a lot of work with computers.
That is currently the case, yeah.
But not
Yes.
always?
No, in the past I've done mostly wet lab stuff, so actually moving tiny little volumes of different liquids around.
Yeah.
So I did like genetic testing in the past, and then R&D for genetic testing, and that was mostly actually doing sequencing.
Oh, okay.
Yeah, we had separate people who do the computer work here, but
Yeah.
I mean there we had, and now here I have to do it myself.
Okay.
But,
Because you're kind of the main person doing molecular biology here, I would say, or the
Yeah.
only person.
yes, Max will be an expert so soon.
Oh, okay,
Yeah.
great. Yeah.
But yeah, here I guess I'm the main molecular biologist.
Seems like a really important part of it. Yeah. And you're mainly on the Wind Cave project, I guess, the same one that Jaden is working on?
Yeah, that's right.
You're doing a PhD? Correct?
Yes.
And so what caused you to choose graduate school?
I tried very hard to not go back to school. I really like actually working, like being out in the real world. As much as I love learning, I prefer actually having a job and having a little more structure to the grad school format. But at least in molecular biology, there's a point where if you just have a bachelor's degree, you can't really progress in your career past that. So I was running up against that point, so I decided to come back, get a PhD, so that I can hopefully progress a little more.
So you went from bachelor's, then you worked a while, and then you went to the PhD?
Yes, that's right.
Okay, great.
Yeah, and I'd really recommend that progression for anyone else in molecular biology. I think it's just it's really nice to get some real world experience, and then I think that makes it a lot easier to get into grad schools. And I imagine once I actually graduate, that'll make it easier to find a job afterwards to have had work experience.
Yeah, I guess you knew what you wanted to study. You had a more stable situation, so it was probably easier to go into it.
Yeah, that is also very true.
Yeah. So the Wind Cave project, what exactly is that?
That's classified. No questions.
Okay, moving on to the next question.
Do you want me to talk about like the whole project or just my part of the project?
Specifically, you're a part of the project.
Okay. All right.
So you used the same samples, right?
Yeah, approximately. I mean, at least we both used the water. My
Okay.
sample's a little bigger than Jaden's sample. Not to brag about my sample. Okay, the Wind Cave project. So I guess I will talk a little bit about the overall project is looking at autotrophic manganese oxidizers, so bacteria that live by, quote, quote, eating manganese. And my part of the project is looking at the viruses that infect those bacteria. Viruses, like traditionally we would think like virus kills things, like it infects a cell and then it kills the cell and releases more viruses out to infect other cells and kill other cells. And they do that. But especially in bacteria, they do a lot of other things that are interesting, like they drive genetic diversity in their hosts. They release nutrients when they kill the host. The nutrients that were in the cell go out into the environment, and they also transfer genes to their host cells. So my part of the project is looking at whether the viruses in these lakes are transferring beneficial genes to their host cells as sort of a way to adapt to the low nutrient environment, since these lakes are one of the lowest nutrient environments known on the planet. So it's a big question about how the bacteria are surviving in there.
Okay. And viruses may be a reason for that.
Yeah, sort of counterintuitive, but...
Yeah, you don't usually think of viruses as being a good thing.
That is correct, yeah. I was very surprised when I started reading about that. But yeah, it's been shown in other low nutrient environments, especially that viruses can transfer beneficial genes to their hosts, and those genes can make the host may be a little bit better adapted to the environment. So like they can transfer genes that like enhance the host's ability to move around or survive shocks, stressful conditions.
Have you discovered any proof of that so far?
Some preliminary proof, perhaps. So I've been looking at DNA that a previous grad student sequenced from within these lakes, and I've been looking at the virus DNA from within the like larger set of all the microbial DNA. And so far it looks like they're like, on the one hand, there's sort of an enrichment in genes that are defense related. So that is sort of a sign that there's like parasitic infections happening. So, you know, the standard virus kills the host tries to fight back against the virus fights back against the host. So there's defense genes that the viruses have. So that suggests that there are those parasitic infections happening, but there's also, at the same time, there's an enrichment in genes that might be beneficial. So like metabolic genes, they're not necessarily beneficial, but they could be. So there's enrichment in those genes, and then there's also some specific genes like motility genes, so host movement, and also like nutrient acquisition genes that these viruses are carrying. So like pretty preliminary so far, but there's some evidence supporting that there might be this mutually beneficial interaction.
Sounds good. So once you figure out how these viruses and bacteria are interacting, transferring genes and everything, what is the overall goal of the project? Or what is your dream? What do you want to come out of it?
That's a good question. This, unlike the other lab members, there's not so much of a real world application, like immediately for this project. So tragically, no patents in my future, I would expect. But like for me, my goals for this project, so cave viruses, there's been essentially no research about them. There's been probably more than 1,000 publications about the cellular life, so like the bacteria and the archaea. But there's been, at my last count, like 5 papers on the viruses, and none of them were any sort of large-scale look at the DNA. So there's like pretty much zero research in this field right now, which is surprising to me because I know the viruses are important in those environments.
Yeah, it seems like an extremely important part.
Yeah, they have a lot of effects on the cells. Yeah, so I would just like to, I don't know, maybe establish like a base of knowledge about these viruses, go from being essentially nothing about like what genes they have, what their diversity is, to having, I don't know, some amount of basic knowledge.
Okay.
Can be built upon.
Yeah. So it's like you're setting a foundation for this too. I mean, it seems really new.
Yeah, that's the goal anyways. We'll see how it goes.
So like I understand your project really well,
Nice.
but it's opened up a lot more doors into like how does this actually work? You can't see microbes, but you can culture them. And how do you do that with viruses?
Yeah, that is definitely a good question. Yeah, so the question of actually physically looking at the viruses, that is a very good question. In, I guess, environmental microbiology as a whole, there is a big issue about actually being able to grow things. So, like, of all the microbes that are out there, there's only a very small percentage that we can actually grow in the lab. And that's true for the bacteria, and that's also true for the viruses. This is, as I said, one of the lowest nutrient environments on Earth. So probably one of the more common adaptations to deal with that is that organisms will just grow incredibly slowly. So like maybe E. coli reproduces, so like the cell divides every like 20 minutes, but like these cells might be dividing like a lot more slowly. So I don't know, maybe they divide once every week or every month or every year. So they're growing very slowly. So it's difficult to culture them. And the viruses, culturing those would add an additional layer of complexity. So currently, probably not going to be doing any culturing. So I'm never going to be a real microbiologist. It's a big field.
You do sequencing with them too.
Yeah.
How do you do that?
That is also going to be difficult. In terms of actually looking at them, we might like filter some out and do some like electron microscopy to just look at the morphology of the particles. So the viruses are a bunch of different shapes, and looking at the shapes can sort of tell you what type of viruses are in there, like sort of in a really large scale. But to look more specifically at what viruses are there and especially what they're actually doing, so like what genes they have, we need to do the sequencing, which is going to be very difficult, I expect, to do. So there's a lot of technical details, but since this is such a low nutrient environment, the cells and also the viruses, there's just not a ton in this water. So whereas in like ocean water, if you want to sequence the DNA of the viruses, you can maybe filter like a cup of water or something and get a usable amount out of there. But we are going to have to filter at least 2,000 liters of water, maybe up to 10,000 liters. So there's a lot of technical challenges that come with that.
Can you make a machine to do that in the cave?
It's classified.
Is that the robot?
Yeah, that's the robot.
Oh, okay. Now it makes sense.
That is part of the robot anyways. Yeah, so what we are going to try to do to overcome that problem is, I guess, working in our favor is, since there's not much in this water, like that's a challenge for us, but it also means that our filters won't get clogged too easily. So the plan is we have 3 different filters in series. 2 of them collect the bacteria, which are bigger, and then the smallest one is last, and that's going to collect the viruses. So we're going to have like a floating platform with these 3 filters and a pump that will pump like 2,000 plus liters of water through the system. It'll probably take like 24 hours to do that. So yeah, so we'll put that in the cave. We will come back out of the cave, and then probably the next day we'll go back in and collect everything and take it back out.
Wow. So
Yeah.
quite a few engineering aspects here.
Yeah. And then after that, we will probably have still just a tiny little amount of DNA. So from there, we'll have to extract it and then do the DNA sequencing, which will be difficult.
Complicated. I would expect so.
Yeah, I hope that it will not take multiple years to get the DNA, but that is unfortunately definitely possible.
Whew. That's scary.
We'll see how it goes.
Oh yeah. Thank you for sharing about your project,
Thank
and
you.
I wish you the best in your research.
I will need those wishes. I expect you to delete all of that and keep only its class.
Yeah.
Just like the intro and then me saying it's class.